Hello everyone, welcome back to our discussion on evolution, origin of life. So in the last session we have discussed about the we were discussing about the biochemical origin of life, on which we categorized that into two different sessions for the sake of our discussion into chemical evolution as well as biological evolution. In chemical evolution, we saw that how the simple organic compounds or simple organic molecules were converted into uh, uh, the micromolecules into macromolecules, and how nucleoproteins were formed, how the other important biochemical elements like our amino acids, carbohydrates, lipids, then nucleic acids, nucleoproteins, etc., were formed, which all of which form the basis of the origin of life. So in this session, we are going to discuss about the biological evolution of life. And in biological evolution, which is a process in which is in continuation with the chemical evolution. So what happens is after the macromolecules were formed, they went aggregation and precipitation in the primitive oceans, forming colloidal particles of organic materials. And these organic materials were termed or were called in two different terms as coacervates by Oparin and as microspheres by Fox. And both this particular structures, which was defined by Oparin as well as Fox, they were having the same features, or they're having the same, almost the same properties. The major difference is that when Fox described such a molecule, he added some of features for this particular structure, which was formed by the aggregation of macromolecules. So there are many, or there are the defined terminologies, or what is defined by Fox as well as what is defined by Oparin. They are having many similarities. The first similarity is that, or the first thing that has to be discussed about this is that these particular molecules, whether it be coacervates, whether it be microspheres, there were molecules which were distinct, or which were having a distinct structure in the particular primordial soup or the primordial oceans. So structure in the sense that the primordial soup or the primordial oceans being aqueous, this particular which was an aqueous solution, which was the major content was water, and this particular materials, or this particular structures which were formed, which was considered to be the precursors of the first life, they were distinct in the aqueous solution. That means they had a distinct structure in aqueous solution. That means they did not mix in water. So the first structures that were distinctly formed were nothing but lipids. So lipids are the ones which is having a distinct structure, or they can stand distinct in this particular aqueous solution. As we all know how oil can be dispersed in water, it floats with a distinct spherical structure in water, and that is the same, or that can be taken as an analogous for this particular structures, whether it be coacervate or the microspheres. So the formation of this coacervates or the microspheres, whatever it be, both were having distinct structures that did not mix with water. That's why we termed or hypothesized the structure to be having lipid, or it was made up of aggregation of lipids. So in chemical evolution, we said that lipids were formed by mixing of alcohols and fatty acids, and those lipids, when lipids were formed, they started to stand distinct from water, and lipids, they started to aggregate. Lipid molecules itself, they started to aggregate, and that aggregation of lipid molecules led to the formation of such structures, which is called as coacervates as well as the microsphere. And the particular process by which coacervates were formed by termed by Oparin as coacervation. It is a process by which they are formed. The process is nothing but aggregation and precipitation. Macromolecules are aggregating and precipitating, so this is the process by which coacervates are being formed. And the formed coacervates or the process by which they are formed is called as coacervation. And another important feature that was given attributed to coacervates by Oparin was they had the ability to absorb molecules in the sense that after lipids aggregate in the form of a droplet like an oil droplet, they started to absorb materials inside, and this particular as said or as hypothesized, these coacervates contained macromolecules in different combination and in different proportion, and this particular the different combination and different proportion is due to the ability of self absorption or selective absorption. Selective absorption in the sense that some lipid droplets had the aggregates of water soluble or hydrophilic molecules. That means in some lipid molecules hydrophilic molecules aggregated. For say if this is for say this is a lipid droplet and these are two lipid droplets. In this one, when a selective absorption occurs, this was having what is called as hydrophilic molecules, and in this one it was having hydrophobic molecules, and that is because of this selective adsorption. This was having hydrophobic one and this was having hydrophilic one, and the two was because of selective absorption of this particular structures which was called as coacervates. So now we'll see what is the main reason for the selective adsorption. So selective adsorption is because of difference in the chemical nature of the coacervates or microspheres which are formed. Chemical nature in the sense, if you see this particular diagram, you can see that this is a type of lipid droplet and this is another type of lipid droplet which is now termed as liposomes as well as micelle. These are two different type of lipid structures or lipid aggregates. You can call them as lipid aggregates, and the speciality of this lipid aggregates is that we'll first talk about liposomes and then we'll move on to micelle. So what happens or what is the difference is that liposomes are structures which are formed of lipids with two fatty acid tails, and micelle is formed of lipids with a single fatty acid tail. So this is the structure of a lipid where this spherical structure is the head, which is usually hydrophilic and that is made up of alcohol, mainly glycerol, and this is the head, and this is what is called as a tail portion. The tail portion is mainly made up of fatty acids, and this is having two fatty acid attached to it, and this is having only one fatty acid attached to it. So what happens here or what can be seen here is that in this particular case of a liposome, this structure is formed of lipids which are having two fatty acid tails. So when such lipid aggregates are formed, that means with the lipids with two fatty acid tails, they aggregate, they form such a structure which is called as a, which you can see here, which is called as a, which you see here, this is called as the bilayer sheet. So bilayer sheet or such a structure, what you see here, such a structure usually does not exist normally. That means it does not have a chemical stability. That means a lipid sheet does not have a chemical stability. Always it will attain, always every lipid molecule tries to attain a structure which is a stable structure. Stable in the sense that always it will try to hide the fatty acid from the aqueous environment. So if they were attaining a structure like this bilayer sheet, you can see that this particular portion, this yellow portion is nothing but the fatty acid tail, this center portion. So whenever such a sheet is formed, the ends of the sheet will have this fatty acids being exposed to the aqueous environment. Such being hydrophobic, they will never get exposed. As a result, it will attain a structure where the hydrophobic sites or the hydrophobic molecules will be hidden from the hydrophilic or the aqueous environment, and if such a, or if lipids with such a structure, that means lipids with an alcoholic head and a glycerol and two fatty acid tail, they usually form a bilayer. That means in this there are two layers of lipids. The white that you see here is the head group, and the yellow one is the uh, fatty acid tails. So they form a structure like this. That means nothing but they bend, and so when bending, what happens is they are attaining a spherical structure, and this is a cross section of such a structure. So this will be spherical, and the entire ball like structure, or the spherical structure, the outer surface will be hydrophilic with the head which is alcoholic, and the center portion will be, being a bilayer, the center portion will be having this fatty acid tail, and the inner portion, imagine a lipid bilayer like this, and this is going to fold. So folding means you just imagine bending a sheet like this. When you bend a sheet like this, you can have this particular area at the outer surface, and this white in the inner surface. It moves inside, just bending that particular sheet like structure, and you try to create a sphere. So that is a structure which is called as a liposome. So the feature of this liposome is that you can see that the inner surface of this is also now white, which is the head group. As a result, the inside is hydrophilic. Okay, and outside, and you are having the hydrophobic lipid tails here, and all the outer surface is also hydrophilic. And coming to the second structure, the second type of coacervates that can be formed by this type is that you can, as we said earlier, the lipid head with a single tail. In such a or if such a structure, or such lipids aggregate, they do not form a bilayer. Instead, they form structure which is called as micellar. That means they form a bilayer is not formed. They form a try to form a single sheet, and they try to fold. And when a single sheath is getting folded, you get a structure like this, where you do not have the inner core. Examine a single sheet. Single sheet means if I draw many lipids, so consider this as separate lipids. So this is having a tail here, this is also having a tail here like this. So here also you are having, and this is also so consider this four as four different lipids, and this is a sheath. And consider these four, I am going to make a circle out of this four. So what happens is, all the head group, they will cover the circumference, and the lipids will be in the inside, the structure which resembles this micellar. Okay, so this is this is the structure that occurs, or this is the structure that is resultant when you have lipids with single fatty acid tail. So likewise, there is a possibility of having, or possibility of forming two different, this is only two different structures. So there might be n number of structures, or n number of structures for coacervates which might have formed. So in this structure itself, we can find the difference. That means because the inside of this particular coacervate is hydrophilic, this particular structure will be attracting and absorbing molecules, or the inside molecule always will be hydrophilic, because inside is hydrophilic. As a result, hydrophilic molecules like small proteins, small carbohydrate, etc. will be absorbed. But in the case here, the core is hydrophobic. As a result, other lipid molecules, other aggregates of lipid, etc. will be aggregating in such a structure, or such a coacervate which might be having a structure of micellar. And liposome like coacervates might be aggregating hydrophilic molecules inside, and our micellar like coacervates might be aggregating hydrophobic materials. So, coming to coacervates as structures we discussed, when Fox described such structures, he also added, or he also attributed some more features to this particular structures, and he called them as microspheres. So these microspheres, they organized, or they are also organized organic droplets with a double layered membrane, means the same thing. They are also having the lipid bilayer, and they also exhibited growth. So here growth is nothing, you cannot compare growth of a particular unicellular organism which exists now. So this growth is nothing but combining different smaller lipid molecules and enlarging its structure. So this can be taken analogous to what we do, take a drop of oil, put it in a glass of water, and shake that particular thing or particular oil drop with a spoon in that glass. So what you can see is that those oil droplets, it will split into smaller droplets, and after some time, if you give or if you do not disturb that particular oil droplet again, what happens is that again it will aggregate and form a sphere or two or more larger spheres. So that is what is growth, which is meant for this coacervate. That means the aggregation here, there is nothing, a physiological growth cannot be attributed for such molecules. Here growth is nothing but increase in size by aggregating of similar molecules. And here division is the same. Here division is what we did for the oil droplets. Whenever an external force is applied, the drop splits into many different components. So that is fission in this case. So here also fission like amoeba etc. Does not occur. Here fission is nothing but because of some external forces, this particular droplet or this particular aggregate, it may split into two or more components, and that is what is called as fission or division in this case. So this is all about the first precursor of life, the first life living cell, the coacervate or microspheres. So they had the same features. uh, you can attribute the same features for coacervate and microspheres, but they are defined by two different persons. Coacervates were defined by or detailed by, hypothesized by Oparin, and Fox hypothesized such a structure, and he coined it microspheres. So coming to the next level, from our coacervate to the other one. So what happens is the first living cell were called or were defined as a protocell. It is called as a protocell, or another one which is a protobiont, and it is hypothesized that this it originated in protocell or a protobiont. It originated in the sea spontaneously, and the particular site where the first life might have originated is hypothesized to be something which is called as a hydrothermal vent. So this is a hydrothermal vent which is seen deep in the oceans. So hydrothermal vents are nothing but they are the particular portions or particular sites in the floor of the ocean, uh, where the floor of the ocean, there are crevices which are found in the floor of the ocean that connects to the, or that reaches to the internal or the depth of the core of the earth. So they are deep and they reaches the depth. So whenever water infiltrates such depth, so in ocean always water infiltrates into such crevices and they reach the core. And because the core is too hot, what happens is the water which enters there, it forms vapors mixed with many chemicals which are present in the core, like hydrogen sulfide etc., and they are bombarded out, or they form as fumes, as you see here, and they are expelled in the form of fumes. And fumes contain all these chemicals like hydrogen sulfide and many other chemicals whatever is present in the crust of the earth or in the mineral rocks which is present here. And it is at such sites, to be believed, or it is believed that it is at in this particular sites which is called as the hydrothermal vents, the first life or the first living cells might have originated spontaneously. So the protocell also, where they also is given the same structure of the coacervates, or they evolve the same structure of the microspheres or the coacervates. They have a spherical, enveloped with a double membrane, and also without a nucleus, and they're reproduced by budding. And being anaerobic, they were considered to be anaerobic because they might have evolved because the first cell is hypothesized to be evolved before the evolution of chlorophyll. As a result, they might be anaerobic, and they might have started to produce energy by the process of fermentation. The prototropes were thought to fed heterotrophically in the organic soup. And from the pre-cell to a living cell, so some chemical reaction resulted in the formation of special proteins and enzymes, and those coacervates which were having inside them. So as I said, in a liposome like coacervate, they might attract molecules which are hydrophilic, like proteins, then nucleic acid, etc., or and they might form nucleoprotein inside and a membrane which is outside, which resembles a virus. So such a structure might be the first living cell with free genes which are united to form a larger one with a limiting membrane, and that is similar to the virus which exists now, and that's why the structures are called as protoviruses. And two major groups of cells might have appeared during that time, the cells with a nucleus and cells without a nucleus. It is considered that because chlorophyll might have evolved afterwards, the first living cells are heterotrophic and with anaerobic respiration, and they might have released carbon dioxide by fermentation. And after the chlorophyll has evolved, which is considered to be a significant breakthrough in evolution, the cells started photosynthesis and started to liberate oxygen, the magic molecule, and that facilitated, or the evolution of oxygen or the release of oxygen from chlorophyll containing cells facilitated the evolution of aerobic organisms, and including both autotrophic and heterotrophic aerobic organisms. And the biochemical evolution, what we discussed in the session, that is the chemical evolution and biological evolution, as collectively was postulated by Haldane and Oparin as a theory which is considered as the Haldane-Oparin theory. So it is same as that of the biochemical evolution of life. They proposed it as a theory which is considered as the Haldane-Oparin theory. So they hypothesized that life evolved in oceans when the atmosphere was a reducing one, containing hydrogen, methane, ammonia, carbon dioxide, and superheated steam with no free oxygen. That's why we consider it as a reducing atmosphere. The organic compounds were synthesized, or the catalysis or the catalytic molecules which were similar to enzymes which function now, were UV rays, were non-biological UV rate and also lightning. The absence of ozone layer and free oxygen to oxidize organic compounds, they were stable and they accumulated in the so-called primordial soup or the Haldane soup. The first living organism would be a few macromolecules wrapped in lipid membrane or lipid. They would absorb from the surrounding. They might have grown, as we said, for our microspheres, and might have obtained energy by fermentation. After the evolution of photosynthesis, they started to feed on the oxygen was released that caused the formation of ozone, and now the atmosphere turned into an oxidizing one, and free evolution or free oxygen led to the evolution of respiration and eukaryotic organism or eukaryotic structures. So, and this is an experiment which is called as Uremilla Experiment. So, Haldane-Oparin theory is nothing but it is the same adaptation of our biochemical theory. So, biochemical origin of life can be it is considered to be in the form of a theory, and that was proposed by Haldane and Oparin. And this particular experiment, which is called as the Uremilla experiment, is an experimental proof for what is called as the Haldane-Oparin theory, or the biochemical origin of life, or you can call it as the molecular evolution. So this is considered as an as a proof for that biochemical origin of life. So what they did was, Urey and Miller, they reconstructed the primitive conditions of the primitive earth in a lab, in an apparatus, and they proved that the simple or the macromolecules could be produced from simple organic molecules, which was proposed in the biochemical origin of life. That means from hydrogen, carbon, nitrogen, and the water vapor, we could get the macromolecules like methane, ethane, etc., and also our amino acids, nucleoproteins, etc. So they proved this particular hypothesis, the biochemical origin of life, using an apparatus which they designed in the lab. And that apparatus was so well designed that they maintained all the conditions that prevailed in the primitive earth. The main condition was, the first thing is that the catalytic property, as we said, the catalysis in such a case, we said that there was no biological catalysis. As a result, UV and electricity was or lightning was uh, acting as a catalysis for all the aggregation, precipitation, etc. So what they did is they provided their apparatus with a continuous supply of electricity, and that is how they simulated. Simulation means they are creating it in the lab. So when you create something which is present in nature in a smaller form in your lab, that is called as simulation. So here they simulated that particular the lightning as well as UV in the form of continuous supply of electricity. And next is the presence of the reducing environment. So primitive earth had a reducing environment. So how they created it was, when air is present inside the apparatus, it will be oxidizing because you are now having oxygen in the air. So what they did is they sucked out all the air from the apparatus and with the vacuum pump and closed it or sealed it. As a result, it was vacuum inside, and that can be considered to be a reducing environment, because now the apparatus will have only whatever gases is provided from by externally. It will not have the external or the atmospheric air which is present now. So now we'll see their experiment setup. So this is what is called as a Uremilla experiment. So what they did here is, so here is the chamber where the electricity was provided, and what they did is they boiled water here for water vapor, and through this one they supplied ammonia, methane, and hydrogen, etc. So all the gases, ammonia, methane, and hydrogen plus water vapor, they mix and they get mixed in this particular chamber, and in this chamber they provided the electricity as you see here. So carbon dioxide, nitrogen, ammonia, hydrogen, etc., they mix here in the presence of electricity. And what they did is, this will be in the form of vapor because this is gas, and they provided a coolant, or this was a glass apparatus. In the glass apparatus, in this particular portion, they covered another glass apparatus, and this was completely supplied, always running with water, not inside the outer jacket. The white tube goes in the middle, and you can see this purple color, that is the outer covering of another glass chamber, which is always containing running water. So as a result, the gas will cool here, and droplets will come. The droplets will be collected here, and they will collect the droplet from here, and that droplets will be analyzed for the contents, whatever is present. So whatever chemical reaction happens here will be present in the droplets, and they will collect the droplets here, and they analyze the droplets with the help of the particular technique which is called as liquid crystallography. They did liquid crystallography for this particular for analyzing this particular compounds, and they found that this particular mixture which was present after they collected after 1 week, they contained amino acids. They contained the simple acids like acetic acid, etc. Acids were present. They contain simple amino acids, and that was the proof for uh, or that was the evidence, or that was a full proof of Haldane apparent theory, or the biochemical origin of life. That means from simple compounds, if we are creating a primitive atmosphere, the compounds which were hypothesized by Haldane in the biochemical evolution of life could result. And some other scientists in also without such an apparatus, etc., they mixed our hydrogen cyanide with some chemicals, and they also could obtain the nitrogen bases like adenine, and these were the proof for proof to substantiate what is called as the biochemical evolution of life, or the molecular evolution of life, or the Haldane operant theory. So with this we have come to the end of this particular session, that is origin of life. In Origin of Life, we discussed about the earlier theories of evolution or the origin of life. That means the earlier beliefs which said about, or which were present, or which were prevailing about how life evolved in Earth, and also we discussed about the present theories like the biochemical origin of life and its Haldane operant theory and its proof, which is the Uremilla experiment. So in the next session, we'll be seeing the other proofs of evolution, like our Darwinian principles, the Lamarckism, etc. So with this, we come to the, or we conclude the session. Thank you for listening.